everyone. Such an honor for me to be here today and talk about the biosignals acquisitions that have been made as part of the Movie 10 dataset. My name is François Lespinasse, and I've been interested in the cardiorespiratory activity of our subjects. So, why does Neuromod actually acquire such data? It's because it's interested in emotions, basically. Emotions or affect are broadly understood to include phenomena such as feelings, moods, and even mood disorders. And globally, research points to the idea that affect might underlie or even influence a myriad of higher order cognitive abilities, such as attention, memory, and decision making. So, keep in mind that these higher order cognitive abilities will be tested as part of Neuromod's large scale scanning experiment. So, we intend to use physiological data to annotate our participants' affective states in order to associate these affective states to fluctuations in bold activity or even variations in behavioral performances. So, where does one stand knowing the value of the data that's being generated and also the ambition of Neuromod to provide high quality data to assess its participants' affective states? We start by defining what emotions are. Basically, we all know that emotions modulate according to features in our environment or in our body, that is, phenomenal features. Russell's explanation was to reduce the dimensionality of our experience to two main aspects, which are bodily and mental. And more recently, this theoretical suggestion has been integrated into dynamic systems model of human neurobiology, and saying that these two are actually entangled and they form a self-organizing complex. And so we might say, in light of recent research in emotion, that whatever the semantic complexity, whether it's fear, sadness, admiration, envy, aesthetic experience, all of the above, all these emotions are tied to lower level processes, that is, implicit processes of the nervous system. So, what's happening essentially outside the brain. So, we might suggest that in order to get a comprehensive picture of emotions, we need a comprehensive picture of the nervous system's activity itself. So, Thayer and Lane inspired this approach. They suggested that measures of autonomous activity are tied to activity of specific brain regions that form the central autonomous network. And this network includes the interior and posterior insulas, cingulate cortex, and amygdalas. Meta-analyses gave credit to this model, reporting that top-down regulation of involuntary bodily activation can be assessed by measuring both brain and physiological activity. And most prominently, this has been done by using heart rate variability. Why heart rate? It's because we know that high frequency variations in heart rate, that is, changes ranging between 2 to 6 seconds, have been extensively used as an index of parasympathetic control of heart rate. That is, one component of the autonomous nervous system control of heart rate. So as low frequency changes in heart rate are tied to respiration, we think that recording this semi-voluntary activity would help us gain a more precise measure of the different contributions of both autonomic branches. So we believe having these indexes of sympathetic and parasympathetic activity would be valuable to assess a participant's affective states. And these features of the activity could be used in further decoding tasks, such as the one from Orikawa 2020, where they decoded emotions in their participants by using the central nervous system's activity. What would happen to this model if we added biosignals to it? So could we gain some measure of the reciprocity between the central and autonomous nervous system? So another inspiration for this approach is work by Valenza, who proposed to use high frequency heart rates variations as a seed for the activity of certain brain regions. And of course, what they found out is that it was tied to activity of brain regions specified by Thayer and Lane's model. But before we get into that, I needed to process the signals. I needed to make sure that my analyses were reproducible. So I been using and contributing to open source community driven tools to make sure that these soon to be published data would follow community guidelines. So let's get into more details about the concurrent biosignals and neuroimaging acquisitions that have been made for the Movie 10 dataset of NeuroMod. So Movie 10 is of course 10 hours of data by subject, and it's recorded throughout approximately 10 sessions. Each session have a variable amount of runs. Each runs represent 10 minutes long of neuroimaging acquisition. So for a total of 61 runs by subjects. Biosignals are actually recorded continuously during this time, so even in between neuroimaging acquisitions. So massive amounts of physiological data are generated, and in fact they span even longer than the neuroimaging acquisitions. In order to deal with such amounts of data, files have to be converted in a format that facilitates its analysis and dissemination. So you might be familiar with this slide, as I borrowed it from a previous presentation by our data manager Basil, because my work has been essentially to integrate the phys2bids converter for the biopack data that we have. After these data management steps, I could use another community resource named NeuroKit to extract information from our biosignals. And final step, we analyze the composition of our physiological activity, and mainly here my interest today is to talk about the cardiorespiratory indices that were extracted. All of this is summarized in my conference poster for Organization for Human Brain Mapping conference. So I would be delighted to hear any comments about this work and discuss my work or the work of the Phys2Biz and NeuroKit's contributors. Here I break down the Phys2Biz pipeline. The yellow box contains the physiological recording file that spans along several neuroimaging acquisitions. And essentially, we take that file, give it to Phys2Biz with the appropriate channel information, the number of volumes by neuroimaging acquisition, and Phys2Biz outputs a BIDS compliant format version of each run. So here's the tree of the Movie 10 BIDS dataset with the source data. We have the physiological data that sits under the source data that can be sent under the other functional data of the appropriate subject and session. Here, what I want to show you is relatively simple, that physiological recordings are filled with human errors. Not only do we have variation in electrode placement and technical problems with the equipment itself, we also have invalid runs that we have to discard. So here the figures show you essentially the raw signals plus the trigger signal, which are represented by the red lines. So each red line shows when a brain volume is being scanned. And so an acquisition with 3 volume is of course to be discarded. Here I go into more details about the pulse signals, or signal of interest, in the application of NeuroKit processing pipeline. So the top axis is in the blue trace is actually the raw pulse signal. Then under it we have a bandpass filtered version of the signal, or the results of the cleaning method for PPG that is peer-reviewed and implemented in NeuroKit by the community. So finally, the bottom line in green shows a smoothed version of the computed heart rate based on peer-reviewed methods that were implemented in NeuroKit. So rate is computed by measuring time interval between detected peaks. You can easily imagine that long acquisitions of 10 minutes and more, are filled with noises, motion noises, interferences that render peak detection very hard. So these challenges can be dealt with the proper visualization tools. Here we can see that the distribution of interval on your right shows the presence of outliers. And so this can be dealt with by using the proper interval dependent adaptive thresholds for peak detection. And we can end up with a distribution plot like this. Here we do the same thing. We break down the processing pipeline for the respiratory signals. So we have a raw signal in blue. Then we know that extremas can be detected on the bandpass filtered version of the respiratory signal. So we can compute rates by measuring the time interval between each respiratory truce, or at the end of each expiration. So indices of respiratory activity, including phase, amplitude, and rate, are computed from Codadad 2018 paper, which is another peer-reviewed method implemented in NeuroKit. So the community is dedicated to implementing all these peer-reviewed methods in the literature. Here is a relatively understudied measures that follows the same logic as heart rate variability. So same as before, the plot shows the relation between successive intervals. The red line of the ellipse shows the short-term variability, and the yellow one shows the long-term variability of respiratory activity. So these measures were recently used in meditation experiments where researchers were able to capture both autonomic branches' contributions. So this is interesting to us because in neuroimaging contexts, respiratory measures were mainly used for denoising, and to our knowledge, only respiratory cycles have been used to regress activity of brain regions. So interesting avenues for research. So let me jump to preliminary results. The scope of the analyses made here are essentially to authentify the biological soundness of the signals themselves and to go over some of the metrics that we can extract from the signals. Let me also repeat the number of data that was generated for the movie 10 dataset. So 10 hours, 10 sessions approximately, and a total of 61 runs by subject. So here is a count plot that helps you appreciate how much data was lost in the process. So noticing how many outliers were generated by our processing algorithms for breath metrics and also heart rate data, we decided that a thorough visual inspection of each session seemed like the most realistic thing to do to keep the most data. So how does it actually look? You can see the gap between subject 1 and subject 6 at the level of PPG. We have to explain this gap in quality of acquisition. So here we can kind of see something that looks like a pulse bump, but even a cardiologist cannot assume anything from this signal. Same thing here, but a little bit worse with high frequency noise. We expect it may be some interference in the circuits, and here we suspect that the equipment itself may have been broken here during the acquisition, and so there's a lot of these error in the process of acquiring physiological data. There's a lot of problems related to acquiring physiological data. Here is a distribution for the mean beat to beat interval extracted from the PPG signals. So there's a count of the number of runs. So the mean interval is computed in each run. So each 10 minutes we have our average heart rate essentially here. And you can kind of see a normal distribution, but you know, comparison between subjects and correlation between subjects are quite unfeasible here because the number of runs don't correspond, between subjects. So, yeah, difficult to analyze. Then we can go over the respiratory signal, which would seem to be reliable even in fMRI context, but we still have some weird noise such as these. I haven't seen that in anywhere else, so if you have any input on the source of that noise, please let us know, because the filter, so you see the filtered version in orange here, integrates some like impulse response that corrupts our ability to capture cycles in the respiratory signal. So here, well, we suspect that the equipment itself wasn't set up all right, so we didn't capture anything from the abdominal. And here we can see that a respiratory cycle is done at each 2 seconds, which kind of seem like hyperventilation, or even like it was a simulated signal. So we suspect it may be in the acquisition system itself. Same thing here. We have a count plot, the number of runs for each subject, and you can appreciate that the distribution is different. Some of the subject exhibits more tail distribution, and others have a more constant respiratory cycle. So here are only 4 subjects, and here are only 5 subjects. Difficult to make more thorough analyses between our subjects. It's easy to conclude how ambitious the project of assessing a subject emotional experience really is. In fact, it's really ambitious to want to assess the emotional experience in the scanner using biosignals. Even though there has been lots of unsatisfying observation made by the team in the recent years for the acquisitions themselves, and even here in this talk, we still believe we've made some serious progress. So, one milestone has been to use and contribute to open source community driven tools for biosignal processing. So, Neuromod benefits from the tools provided by the community, and the community can count on Neuromod to provide high quality data to test the developed software. So apart from these corollary advancement, where we believe this synergy between community will forge strong, long-lasting solidarity in science. Apart from that, we still assess the quality of the biosignals themselves. We've concluded that there was a lot of human errors in the process, and it points to the need to establish community-defined, best practices for all of the steps I mentioned here in my talk. So, lastly, we can still use some of the data, and there is ongoing discussion on how this data will be provided to the public. What are the next steps? Essentially, provide even more quality control for the cardiorespiratory indices, specifically for the heart rate variability indices. Here I linked the NeuroKit 2 website, where we can find an ongoing study where we analyze the statistical structure of heart rate variability. We plan on adding Neuromod data to these analyses. Then my goal in the next months will be to regress bold activity, so brain activity, with HRV, so high frequency variations in heart rate. This will be done using the point process analysis proposed by Valenza, and we think this could highlight some interesting biological information on the interaction between autonomous nervous system and the central nervous system. And lastly, I would like to extend the workflows to other Neuromod data sets, beginning with friends. So yeah, there's a lot of work to do. I have to leave you guys, but before, thanks to my wonderful team, thanks to my supervisors, both Pierres. They've been supporting me, going out and reaching out to communities. And thanks to my colleague Jan Arel, who provided awesome feedback. And lastly, but not least, the NeuroKit 2 and Fistubits contributors. You guys made it, and I believe there will be fantastic things going on for us in the next few years. So, thanks again, everyone, and see you soon.